Okay. So yeah, originally I was going to talk on a different topic, but through the events of the past 6 months and looking at the advance of ThryvOn technology, which is now being planted in Arizona, decided we really ought to have a concentrated session just on ThryvOn cotton here. Really haven't spoken about it much as we've been working on it over the years, and I've been working on it for over a decade with Monsanto and now Bayer. But now that seed contracts are out and other programs for planting these acres are starting on a small scale this year and a larger scale next year, I thought it was important that we review the technology and try to answer some of these frequently asked questions. So I'll just say right up front, the key question really is, do you need to control ligase and ThryvOn cotton? And if yes, when? And I'm going to dodge that for the moment, but I'm also going to tell you that that specific research has not been done yet. That might be a little bit surprising to you, given that we've been working on this technology this long, but you need to recognize that an awful lot of development goes into producing a new genetically engineered trait. And that was no different here. So we never had the luxury of being able to pursue some of these questions because we were busy screening, trying to identify and screen the best trait, optimize it, identify its efficacy, basic efficacy and spectrum of activity and safety in the field. And sadly, we don't have a specific trial where we can point to a very specific action threshold or a timing for when and if you should be spraying ThryvOn cotton. But that doesn't mean I'll dodge the question completely. We'll touch on this a little bit later. Before doing so, you really have to back up. And I use this pyramid metaphor all the time. And it's important because we need to remind ourselves, what is our strategy? It's really based in these three large keys of sampling, effective chemical use, and a base layer of avoidance and prevention tactics. And that's no different here. And the trick here is to make sure that the ThryvOn technology is compatible with the successful IPM that you guys have been practicing really for decades now. And it's also important to identify what it is. It's the cornerstone tactic of any IPM plan, which is tolerant and resistant varieties. What we have here is a tolerant variety, whether it was conventionally produced or genetically engineered. Tolerant and resistant varieties are cornerstone to IPM programs because they reduce the impacts of the pest insects. And that makes everything easier, right? It makes all of our tactics work better. It saves you money and hopefully protects against any yield loss. And ThryvOn technology is in fact a tolerant variety. In this case, it's a genetically engineered variety, and genetically engineered with a BT trait. Now, you may be used to hearing about BT only in terms of controlling caterpillar or lepidopteran pests in cotton, such as pink bollworm, which is now eradicated. But BTs exist in many different brands, thousands and thousands, in fact, that have the potential to control different arthropods. This particular one, Cry51AA1, was screened and optimized by Monsanto and sourced, like all BTs, from a common soil bacterium. So they took the genetic material for Cry51AA1, which was known to be toxic to ligase, and they put that gene inside of the cotton plant. This is a picture of that Cry51AA, that protein, that toxic protein. And at the outset, they really thought they were only going after a ligase control material. We've learned since that it has a different spectrum of activity than just ligase. So what are the targets of this technology? That's an easy one to answer. What will ThryvOn control? Well, basically several species of thrips, all belonging to the same genus, Frankliniella thrips. And you need to know there are many, many types of thrips in agriculture. Not all of them are in cotton, of course, but there are many in agriculture, and only a few of them are real serious pests. Frankliniella thrips are important here in the West as they are in the South. two different species usually involved. We also target with ThryvOn ligase bugs. And I'm sure most of you know that there are different species of ligase. We have ligase hesperus in the west as our main cotton pest, but back east it's ligase linealaris. This trait will control or will impact both of those species. So here's what it looks like. You have a cotton plant. Ligase bugs are our key pest that's controlled by this technology. And Western flower thrips are also controlled by this technology, but at best they're considered secondary pests here. It's an unusual day when most growers would spray a thrips population in cotton. Generally speaking, we have good planting conditions and fast growth in our establishing stands, such that what little thrips damage that we do have is easily outpaced by the growth of the plant. And generally speaking, we don't spray for Western flower thrips. So we would definitely consider it a secondary pest in this system. But let's start with that one, because as it turns out, the gains in Western flower thrips control, or Frankliniella species in general, is quite strong. First and foremost, Western flower thrips that grow up on ThryvOn cotton live half as long. So you can see there that they only live about 6 days as an adult versus 14 days in non-BT cotton. So it's shortening their lifespan because they're just not growing as well. And in fact, only about 7% of Western flower thrips make it to adulthood. So it's a good, strong technology for Western flower thrips. Those same thrips, whether they are reared on the plant or arrive in a field, will lay 85% fewer eggs on ThryvOn cotton. There's actually a non-preference or an avoidance that this insect behaves in avoiding any oviposition. Not any, but most of its oviposition on ThryvOn. So if there's an alternative, even if it's a weed patch in your field, they would much rather lay their eggs in those weeds or in adjacent areas than in the ThryvOn cotton. So that's pretty novel. We don't have a BT trait that's like this. There isn't an equivalent. This is a new way of identifying an impact on a pest, potential pest. Also, those that do grow on ThryvOn are 20% smaller. This is reflected in their slower growth as well. So there's really an array of effects that occur, and all this excellent work was done by our collaborators in NC State University, Anders Husseth and his colleagues, and they've shown quite convincingly that ThryvOn is a very good thrips control agent. So in summary, Western flower thrips live half as long, they lay fewer eggs, and they're smaller. And that combination of impacts really reduces the risks for us, what is a secondary pest. So what was a low risk is lower. And in fact, that should make it easy to answer this question. Do you need to chemically control thrips? Well, again, that combination of impacts reduces the risk. And where we rarely spray conventional cotton as it is for thrips, we should be spraying ThryvOn cotton even rarer. I would hate to go on record as saying you should never spray ThryvOn cotton for thrips, but as minor a pest as Western flower thrips are, I think it would be the rare exception that you would ever consider a foliar spray for thrips in cotton. Now, this next point, you're going to see all kinds of press and other results from around the belt that will suggest that others will be recommending seed treatments along with ThryvOn cotton for control of thrips. I can see no rationale for that whatsoever here in the West. So if you are purchasing seed treatment upgrades, that would be a neonicotinoid like imidacloprid at an insecticidal rate. In my opinion, you can save that money, pocket that money, don't pay for those upgraded seed treatments. They're not necessary. You're subjecting those thrips to unnecessary selection for resistance against those neonicotinoids. You're also impacting other elements of the cotton food web, which I'll cover a little bit later. So on the chemical control for thrips, it should be the rare exception that you would either deploy a seed treatment targeting thrips or a foliar spray for thrips. All right, so is the story the same for ligase? Not exactly, but we are still learning, and I hope you can appreciate that we were constrained in all the years that this was a regulated trait to very small plot work. The largest plot we've ever worked on is 18 rows on this 18 rows by 60 feet, which is a good sized plot and is generally reflective of commercial conditions. But with all the unusual ways that ThryvOn can work, I'm going to feel a lot better after we've had a year where we've had these commercial size fields out there. And I would love to work with any of you if you're willing to work with us in monitoring some of these pest populations and tracking them, particularly if you have a non-BT or non-ThryvOn BT field, companion field planted nearby. So ThryvOn trait does target ligase bugs. It does target our ligase hesperus, and it does reduce their impact. We can say that for certain. And I decided I was only going to show you one piece of data, but there's a lot wrapped up in this piece of data. This is data we did here in Arizona, and this is a chart of ligase nymph days over time. So if you're used to thinking about degree days and the way we accumulate temperatures, think about how temperatures accumulate through time and the curves that we could draw from accumulated degree days. This is nymph days. So ligase nymphs are the damaging stage of the insect, the biggest damaging stage of the insect. And here's what the control looked like in 2019 in our experimental plots. We sprayed August 19th because it crested over our threshold of 15 total ligase and 4 nymphs per 100 sweeps. And here's our ThryvOn cotton. And you can see it's a different pattern. In general, it seems to be delayed and smaller or lower than the other. And in fact, we delayed our spray. We used the exact same threshold, which I wouldn't recommend, by the way, but by protocol, we use the exact same threshold for both lines. And we sprayed a little different interval here, but it was a 7-day savings. In other words, it was 7 days difference between when the control line reached 15.4 and when the ThryvOn line reached 15.4. So what does that all mean? Well, this whole area here, this hashed area is all savings to you. It's could be sub-threshold savings. It could be threshold level savings in potential damage to your crop. And that number is 40% fewer or lower than the control. So it's a significant savings. And I think it's definitely going to save on the need for a grower to spray this ThryvOn cotton. And it does this by slowing the growth of these nymphs, particularly the small nymphs. The small nymphs are most susceptible to this toxin. So it can kill some of these small nymphs and definitely slow their growth. That reduces their numbers. And the combination of the lower numbers and the less feeding that results, you end up with reduced damage. And the combination of all of that is really going to equal savings to you when you're deploying ThryvOn cotton. So the key question I started with, do you need to control ligase or really chemically control ligase in ThryvOn cotton? Again, I'm going to hedge a bit. I'm going to tell you that we don't have the experimental evidence for that exact question, but we do have some information. But I have to say, it depends, right? That's the hedge response. But I think I can confidently say that if you're at or below our standard threshold of 15 total ligase and 4 nymphs per 100 sweeps, definitely don't spray your ThryvOn cotton. There's no need to. And if you're at that interval that we broadly reflect as our threshold of 15.4 to 15.8, I think, again, perhaps not. I think that's probably still too reactionary. And once you get above or on the outside of our highest threshold of 15 total ligase with 8 nymphs per 100, I think only then might you consider spraying the cotton. But even still, I'm not confident that the numbers that you will find in ThryvOn cotton are correlated with any predictable yield outcome. By that I mean is with conventional cotton, I can show you very convincingly that 15.4, 15.8 is protective of your yield. We have modeled that relationship over and over again. It's almost a straight line that will show you when you're at these nymph levels and you spray at those levels, it's protective. And if you don't allow those nymph levels to exceed 15.8, you'll suffer yield damage. We don't see that same relationship with ThryvOn cotton, and that only makes sense because the numbers you're counting probably includes both healthy and sick ligase, ones that simply aren't, I hate to use the word, but aren't thriving. They aren't growing well. They're not growing as fast. So they're sickened and probably not feeding as well. So what that means is I think this puts a burden on scouts and PCAs to start looking at the plant more directly. Look for abscission, measure your abscission rates, your fruit retention rates, look for flared squares in your sweet net, maybe do some counting up towards the top 5 nodes, get a sense, and I know all experienced PCAs know what a flared square looks like and what ligase damage, how that relates to flaring of squares. That's, I think, going to be important in understanding whether you're at risk of any losses in your ThryvOn cotton. I can't give you an exact number. I think it would all be related to baseline. So you'd really want to look at the fruit retention curves for cotton. We have fruit retention curves for upland and Pima cottons. And if you're falling below baselines and you can see flaring, I think that's a signal that you might have to supplement with a spray. I would also go so far as to suggest that after that, everything is normal. In other words, I don't think you have to do anything fancy or different. I think you're going to rely on the same 2 products that most of you rely on, which is carbine or transform. Those are good products. We have used both of those in ThryvOn cotton and they work well. So it doesn't change anything once you decide to spray, but I think the decision to spray you need to take much more seriously and take more time to do it because you have more time to do it. Have a discussion, grower and PCA together, and decide, is this growing off the way we want it to? And then come to a conclusion and decide if a spray is needed. In general, in our experimental work, we do see a savings of about 1 spray on average. But again, we haven't done the definitive threshold studies yet. So another key question really revolves around our IPM plan overall, which I've told you many times depends on the beneficials. So is it safe to beneficials and natural enemies? They are central to our avoidance layer in our pyramid. So in short, I'm just going to say yes with an asterisk. I mean, yes, we know, for instance, there's great honeybee safety. There's great safety to the widest array of natural enemies present in our system. There have been no measured negative effects on the majority of the natural enemies in the Arizona cotton system. But we have to review this food web again to sort of understand why I put a little asterisk on that. So this is the food web, right? Cotton's at the base of the food web, and those orange arrows, you can translate those as is fed upon. So cotton is fed upon by ligase. It's fed upon by whiteflies. You know those to be our key pests. Our secondary pests are mites and western flower thrips. Cotton is fed upon by both of those species. But you'll also see these 2 arrows indicating that thrips, in fact, feed on whiteflies, and they also feed on mites. Mites are fed upon by thrips, and whiteflies are fed upon by thrips. So western flower thrips plays this dual role in our food web as both a plant feeder and as a predator. And there are certain times of season when I think it's a very potent predator and probably is spending more of its time helping you out than ever hurting you. But then there are key predators. These are predators that we've identified through research that are very important, especially in managing whitefly populations. So crab spiders, big-eyed bugs, minute pirate bugs, lacewing larvae, Trapetus flies, and Colops beetles. Those are the 6 groups that are driving the biological control that we need for whitefly management. So they're very important, and they probably play some role in ligase biological control as well. And then we have other predators. These are our larger predators like assassin bugs, damsel bugs, Then we have this one, which is known as Rhinocloa and has an orange nymph. These are all good predators as well. They haven't been identified as key in whitefly management, but sometimes they're implicated in ligase control as important biocontrol. The zealous, in fact, is feeding on a ligase nymph. So the question is, what are we doing to this food web, especially with this dual role that Western flower thrips plays as both a potential plant pest and as a predator? Well, we can shrink them, right? We can symbolically shrink those thrips and say, well, we're kind of knocking them out of the system. We saw good data to suggest that you're going to see much lower levels of Western flower thrips in your fields. You're also going to see lower levels of ligase. Well, what does that do? Does that put more pressure on the plant from mites because they're released from predation by these thrips? Does it increase the risk of whitefly infestations because you no longer have no longer have Western flower thrips functioning as efficiently as a biocontrol agent? These are the things that we don't know, and we've been researching for the past 3 years. We're completing that work now. We don't have it done, but I can at least tell you in broad strokes that it looks like, because we have such a nice food web full of membership by many different species, it looks like there's compensation that must be occurring because we're not really seeing increased risks of mite infestations or whitefly infestations as we feared we might. That's good news because certainly we don't want to adopt a technology or depend on it too heavily if all it's doing is increasing the risks to a primary or a secondary pest. But so far, we've not seen any large effects of that, but we're continuing to delve into the data. I'll have more information to share with you by the end of the year. So I'm going to finish with some pictures that illustrate that food web. If you don't like the symbolic nature of that, let's look at this more literally. Here's a picture of an adult thrips. They're very small. These are the wings. This is Western flower thrips. Here are the larvae. These are really important for scouts to be aware of. PCA should be looking for the presence of those larvae because they're the ones that will really key in on those spider mites, and in particular spider mite eggs. These are the earliest colonizers of our cotton fields. Whether you ever have managed them or applied controls for them before, these are the first things that arrive in our field. They actively come off of alfalfa, melons, small grains. These Western flower thrips will migrate readily to cotton, as will mites. Again, they're there almost from the start. As soon as cotyledons are coming out of the ground, these are the species that are colonizing, right along with whiteflies. Now, you never think of whiteflies as being a pest at the seedling stage because they really aren't. But what they are is prey. They are prey items and help recruit all the natural enemies to your field. Without these prey items in your field, there wouldn't be much incentive for these predators to colonize your field. So really, mites, thrips, whiteflies are all important in establishing this food web in cotton because they attract the predators that will give you longer lasting control. So, I think it's prudent, and certainly we saw evidence of this year. This is a picture from this year. This is a picture from about 10 years ago. Both of these from conventional cotton, and they have these what I'll call bruising. Especially on seedling cotton, on those first few leaves, you see this kind of bruised appearance. That's classic mite symptoms there. If you flip that over, you will see the developing colony underneath, and it's been there a while in order to create that sort of scarlet color that you see on these leaves. But whether you've scouted them actively or not, they often resolve on their own. And that's again because of the very active biological control that's being performed by Western flower thrips. It's also by early season lady beetles. Sometimes you'll see lady beetles colonizing these seedlings, and they'll just wipe out those mite populations pretty quickly. So the question is, now that you're tamping down thrips with ThryvOn cotton, will this cause more incidents of seeing these colonization events in your cotton? Now, I can't answer that question directly, but we did observe it this year. We can certainly see it in both conventional and ThryvOn cotton this year. In fact, this picture is from Coolidge It's just shot through a loop, so it's not the perfect one. This is from 10 years ago. This shows the webbing underneath. It's kind of got lots of dirt and dust in there as well, but spider mites do produce webbing along with their colony. There's one single mite here in otherwise what would have been a fairly large colony on the underside of this leaf in Coolidge, if not for the activities of these thrips. Well, here's one. They're kind of a hot dog shaped larva. Here's another one. There's the rest of them. Actually, they're not all well in focus. This one's pretty good, number 4, but there are actually 9 thrips larvae there. And they basically had completely wiped out this mite colony. The only mite on this leaf was this single adult right here. So again, this was non-ThryvOn cotton, but this just tells you the conditions were right this year for mite development in some areas, some localities. And in those areas, if you have ThryvOn cotton, you should probably be paying additional attention, flipping some of those leaves over and looking to see if they've been colonized successfully by mites, or if there are in fact thrips responding and taking care of them. So that's all I have to share with you today. I could probably do a better job answering your questions if you have any now, but we will also put in the chat box basically a write-up of what I covered today that we just produced and are releasing today. I think Naomi will put that in the chat box, both as a clickable link, so you should go off and look at that, or as a PDF. And of course, want to thank our supporters and our team that helps do all this this work. Any questions?
Okay. Thank you, Peter. Any questions for Peter? You can like unmute yourself and ask the question if you want, or if you like, or would like to go into the chat box and write the question, either is good. Any question for Peter so far?
And you'll see Naomi has placed that URL, that web link. You just click on that and that'll bring up the publication that we're releasing today that covers many of these frequently asked questions. I would certainly love to work with any of you, particularly those of you in Pinal County, Maricopa County. If you have ThryvOn out there, we'd love to get out there and monitor the ligase populations as they develop, maybe the whiteflies as well. And if you have a companion field or a nearby field planted about the same time, we could do a nice comparison. If we get enough of those, I think we could learn a lot and share the results with each other at the end of the year.
Okay. Thank you, Peter.
Thank you, Hyman.
Next, we'll have Pedro Andrade, and he will share with us his finding on his work with